metals, and closer proximity to the sun. This no doubt incites question and curiosity about how such an environment can host and sustain life in the case of the pale ale species. However, through many physiological adaptations, this creature thrives in a very unique ecological circumstance. Beneath their transparent gaseous exterior, gas planets contain more complex centres of different elements, such as ammonia, methane, and water vapour. The pale ale encounters little difficulty given its specialised function of water acquisition and storage, in addition to other qualities enabling its inhabitants on gas planet Jovus. Such examples are explored in further detail throughout the presentation. Due to the depth and therefore pressure applied on such gases towards the nucleus of these planets, they appear to behave more liquid-like, which can be observed in the case of liquid metallic hydrogen. This is seen in the bottom diagram of this slide. In a similar way, pressure changes as a result of atmospheric storm activity determines oxygen availability on Jovus. Specific altitudes yield limited amounts of oxygen, which is an otherwise deficient resource in gas planets. The pale ale's specialised flight ability assists in the retrieval of oxygen when the opportunity arises. The many integral and physiological characteristics possessed by the pale ale species facilitate its survival and suitability to the harsh surrounding conditions of gas planet Jovus, in the absence of what we would typically classify as a flourishing ecosystem on planets like Earth.
Due to its low oxygen environment, the pale ale has developed many respiration adaptations, one being anaerobically producing energy. Pale ale has adapted to survive low oxygen environments through the use of anaerobically metabolizing fructose, that of which it gathers from its frugivore diet. This process allows the creature to survive up to 5 hours when it is required to go beyond the oxygen-rich layer. This adaptation helps the creature survive as it is able to produce energy through anaerobic processes that allow it to maintain its metabolism. The adaptation is used more as a backup when the creature is migrating between oxygen-lacking altitudes. Another respiratory adaptation this creature has developed is its considerably sized lung-like structures in comparison to its body. This allows the creature's lungs to have more surface area and therefore more alveoli that can diffuse more oxygen when the creature is at an oxygen-rich altitude. The oxygen, once diffused into the body, is then stored into the creature's blood and muscles due to the fact that this creature has a large amount of oxygen-storing proteins, such as myoglobins and hemoglobins, in both their muscles and their blood, allowing it to have oxygen available when even at an altitude that is lacking oxygen. This is the creature's primary source of respiration. In order to account for the atmospheric pressure changes in this creature's environment, the pale ale has developed chest structures that have the ability to passively compress the lungs when needing to adapt to the different atmospheric pressures. This is in order to avoid the excessive uptake of nitrogen. Without this adaptation, the creature would be at risk of what we know on Earth as decompression sickness. The pale ale can actively control the opening and closing of their tracheal-like tubes and oxygen storage sacs when they ascend to altitudes of less oxygen. They can constrict these passages to minimize oxygen consumption. Conversely, when they encounter the oxygen-rich layers, they can dilate their tracheal network to absorb more oxygen.
We have spoken about pale ale's respiratory adaptations and enhancements. We now need to cover how pale ale's circulatory system transports oxygen around the body. Firstly, we will cover pale ale's kidneys. Pale ale has developed highly sensitive kidneys to detect the most minor changes in erythrocyte levels within the bloodstream. This is to combat pale ale's challenging atmospheric conditions. When the kidneys detect a drop in erythrocyte levels, the kidneys release erythropoietin to stimulate erythropoiesis within the bone marrow. Furthermore, Pale ale has developed methods of enhanced oxygen transport. The circulatory system has a high flow rate, capable of rapidly transporting oxygen to tissues. Vasoconstriction redirects blood away from less critical tissues and organs, ensuring that the limited oxygen supply is prioritized for the most vital areas. Vasodilation does the opposite, ensuring the blood flow is directed to the muscles and organs when most required. Vasoconstriction is the contraction of smooth muscles causing the constriction of vessels, while vasodilation is the relaxing of the smooth muscle of the vessels causing them to dilate. This ensures that oxygen-rich blood can reach tissues quickly, even when oxygen concentrations in the atmosphere are low. These are essential in ensuring pale ale is able to regulate its oxygen to specific parts of the body when required. Pale ale's heart possesses enhanced contractile strength, enabling it to pump blood with greater force. This ensures that blood is ejected into the circulatory system with enough pressure to reach even the farthest reaches of the body. Pale ale is also able to quickly adapt its heart rate to the specific atmospheric conditions.
The surface skin of pale ales possesses an extraordinary and multifunctional cuticle-like structure inspired by complex principle of superhydrophobicity. This unique skin adaptation allows it to interact dynamically with water, adapting its properties based on environmental conditions and its needs. The pale ale features an intricate arrangement of microstructures, including nanotips, lamellae, and ridges, nanostripes, as seen in part A and B of the image respectively. These structures play a great role in controlling the interaction between the skin and its environment, enabling it to switch between distinct contact mode and adhesion levels to moisture, or perhaps toxic gases that may condense on its skin. In certain scenarios, when the animal is exposed to water vapor, pale ales adopt so-called composite contact mode. This mode involves the skin microstructures allowing the liquid to contact only the tops of asperities while trapping air in sub-micrometer scale hollows. This arrangement minimizes the contact area between the skin and the liquid, resulting in superhydrophobicity. This mechanism is especially advantageous when the animal needs to repel undesired gaseous elements or compounds. Conversely, when pale ales requires water, the microstructures undergo a transformation. The nanotips and lamellae closely align, causing the water vapor or fog present in the surroundings to make a wet contact. This increased solid to liquid contact area forms a nearly continuous contact line. In this state, the animal experiences a high adhesive force, allowing it to pin liquid droplets to its skin surface, even against strong gravitational force or during high wind events. Pale ales can effortlessly switch between these contact modes and adhesion level by changing the orientation of their flexible nanotips and microscales. When the scales are tilted flat, the microscale and nanotip separate and favor the composite contact, which facilitates droplets and gas rolling over it. However, when tilted upward, the close arrangement of these structures promote a wet contact and strong adhesion level. This control over its interaction with surrounding elements makes the pale ale well equipped to thrive in diverse and challenging environments. Underneath this advanced epicuticle, the epidermis of pale ales exhibit a remarkable adaptation for water collection. This specialized skin layer serves as a highly efficient water harvesting system for survival in their unique habitat. It is The endocuticle and epidermis that features an array of tiny spines, bulbs, and trichomes, meticulously arranged across its surface. These structures work collectively to collect and retain water from the surrounding environment. At the core of this water collection process are the spines, each adorned with oriented bulbs. When pale ales go through water clouds, water droplets adhere to the bulbs, creating initial deposits on the tip side of the spine, as seen in part A of the image. As water continues to accumulate, these drops merge, forming a larger drop that moves toward the base of the bulb. The skin's ingenuity doesn't end here. The drops collected on adjacent spines are directed toward the base side of the spine, which is covered with trichomes. When water drops contact the trichomes, they are rapidly absorbed. This rapid absorption process ensures the replenishment of the organism's water reservoir, critical for its survival in drier conditions. The trichomes present on the surface of pale ales are remarkable micro and nanoscopic wing-like structures positioned at the base of the spines, as seen in the picture. These trichomes serve an important role in the absorption of water, which is vital for the organism's survival and growth. These structures are highly hydrophilic by nature, which means they have an innate affinity for water. During wetting events, the water is directed toward the central foot cells of the trichome by a simple osmotic gradient, constantly held by dihydrogen monoxide, or H2O, pumps found further inside the organism. A notable feature of this water uptake process is that it is unidirectional, as the lipid-rich base of the trichomes acts as a barrier to prevent the counterflow of water or its premature evaporation when the environmental water potential decreases. Underneath the trichomes is a vascular tissue responsible for carrying the absorbed water. It is made of cells so big that we will refer to them as fibers. Those fibers are highly adapted to carrying water through the use of dihydrogen monoxide pumps that lines its membrane. To fuel such energy-intensive activity, we discovered that those fibers are packed with mitochondria. Mitochondrion are the powerhouse of the fibers and are responsible for generating ATP through oxidative phosphorylation. We observed that on average, each fiber contains 8,000 mitochondrion. That differs from what is found on Earth, in the sense that the mitochondria of pale ales do not conform to the structure of an envelope enclosing an inner membrane, but rather are just made up of highly infolded pellicles. These pellicles have a large number of tube-like invagination or cristae and are the sites of various respiratory chain components and ATP synthetase. We are unsure why the mitochondrial structure of pale ale lack an outer envelope, but we suspect it has to do with the efficiency of delivering ATP to the water pumps.
Once water has been absorbed from the nanotips and lamellae, the water is transported through channels to reach the specialized organ, which is similar to the small intestines of Earth-like mammals. This organ helps to filter water, but is not a direct part of the digestive system. Pale ale digests other foods orally, but the two tracts converge during excretion. The specialized small intestine-like organ can filter water molecules that have been collected. The villi and microvilli that cover the surface of the organ can help filter the molecules. These are finger-like structures which increase the surface area of the internal wall of the organ, helping to increase the volume of water which can be processed. Larger molecules will not pass through as they are made up of a complex compound, and they will not fit through the channels, while smaller water molecules will pass through as the organ is specifically designed for this. Contaminated molecules are filtered down the intestine to the ceca before being expelled. Larger particles are expelled with other waste products, turned into a uric acid, similar to Earth-like birds. The ceca is the last area for water molecules to be collected before excretion. Pale ale does not have a bladder, which makes it lighter as it does not hold any liquid waste or have the weight of the organ. The ceca acts as a final catchment in this system before excretion. Pure water molecules that may have been missed in the first stage of this process have a second chance at uptake before being expelled. This helps to maximize total water uptake. The refined water which has passed through the villi and microvilli then follows the osmotic gradients of the creature to serve where best needed. Pale ale can store purified water in the enlarged vacuoles it possesses, but this reserve is only used when in areas without water vapor, which is not where the creature typically tends to be, which is why it does not possess a storage sac or hump.